Hello, everyone, and welcome to our module on the blood-brain barrier. So the blood-brain barrier is a barrier that surrounds blood vessels, especially capillaries in the central nervous system, so regions of the body like the brain and the spinal cord. And it controls the content of the central nervous system interstitial fluid. Only some things can cross this barrier, many things cannot. There are a couple of different elements that make up the blood-brain barrier, but two of the important ones are, first of all, tight junctions between endothelial cells. So these surround capillaries and they limit the movement of substances into brain tissue. Also, astrocytes in the central nervous system have foot processes, and these terminate in an overlapping fashion on capillary walls, so they also contribute to the blood-brain barrier. And so in this video, we'll spend some time talking about how different substances can move across this barrier, which substances can cross it, and which cannot. So first of all, some substances that easily diffuse across the blood-brain barrier are shown here. So first of all, water, also gases like oxygen and carbon dioxide. Ethanol easily crosses the blood-brain barrier, and that's why when people consume alcohol, they can have central nervous system effects from that. And then finally, small molecules, especially if they're lipid-soluble. Lipid-soluble substances much more easily cross the blood-brain barrier than other substances. Things that cannot easily cross the blood-brain barrier include anything that is not lipid-soluble. So anything that's polar or charged is going to have a difficult time getting across the blood-brain barrier. And this is the case for some drugs. They're unable to penetrate the brain tissue, and that's because they aren't lipid-soluble or they are charged. Also, other bad things are kept out by the blood-brain barrier, so like bacteria and some drugs and toxins that could be harmful. These things are kept out because of the blood-brain barrier, and that's probably why it evolved. Two special things to know about are glucose and amino acids. These substances cannot directly cross the blood-brain barrier, but obviously our brain cells need glucose and amino acids so they can get into brain tissue, and there are carrier-mediated transporters that are able to pull them across the blood-brain barrier. So now what I'm going to do is talk about circumventricular organs, or CVOs. These are vascular brain structures near the ventricles of the brain. That's why they're called circumventricular. And they have highly permeable capillaries with no blood-brain barrier. So these structures allow direct communication between the central nervous system and the bloodstream. Some of them are sensory, they're designed to sense things in the bloodstream. Some of them are secretory, they're designed to allow the brain to secrete substances into the bloodstream. And there are four key CVOs you should know about. They're all listed right here, and we'll go through each of them one at a time in the next few slides. So we'll start by talking about the area postrema, which is found at the caudal end of the fourth ventricle in the medulla. So let's look at this drawing on the slide here. So this is the brainstem right here. This is the cerebellum shown right here. And in front of the cerebellum, between the cerebellum and the brainstem, you have a ventricle. You have a space in the brain that contains cerebrospinal fluid, and that is called the fourth ventricle. And this is where you find the area postrema. It's on the bottom end or the caudal end of that fourth ventricle. And what you find in the area postrema that's clinically relevant is the so-called chemoreceptor trigger zone. This is a region of the central nervous system of the brain that's outside the blood-brain barrier. And chemotherapy agents used to treat cancer affect this area, and it sends signals to the vomiting center of the medulla. So this is the reason why patients who are placed on certain types of chemotherapy drugs often develop nausea and vomiting. Those drugs can't directly penetrate the blood-brain barrier, but they're sensed by the area postrema, which is outside the blood-brain barrier. Okay, our next CVO is the OVLT, which stands for Organum vasculosum of the lamina terminalis. And it gets its name because organum vasculosum means vascular organ, and it's near the lamina terminalis, which is a thin membrane found on the front wall of the third ventricle of the brain. Remember, all of these structures are going to be near one of the ventricles. This is the OVLT shown right here in this image. It's on the anterior or front portion of the third ventricle. And the key thing that you find in the OVLT is the osmosensory neurons. So there are neurons here that can detect the osmolarity of our blood. And so this is important to tell the brain how much sodium and solutes are in our blood so that the body can regulate blood osmolality. And I talk a lot about this in the renal videos about how antidiuretic hormone maintains sodium concentration and osmolarity. But what you want to know here in respects to the central nervous system is that these osmosensory neurons that detect our osmolarity are found in the OVLT. Okay, our next CVO is the subfornical organ, also called the SFO. This is also found near the third ventricle, but in a different spot from the OVLT. So as you can see in this drawing, the OVLT sits here, right above the optic chiasm, not too far from the pituitary gland. The SFO sits up here. It's often described its location as the roof of the third ventricle. It's right below an unusual structure called the fornix. That's why it's called the subfornical organ. And what the SFO does is it responds to many circulating substances. Its exact role is not clear. The thing about this CVO that's best described in the literature is that it can respond to angiotensin II. There have been studies where they infuse angiotensin II into animals, and it's detected by the SFO, and from there other things happen, like the animals become thirsty or their sympathetic nervous system gets activated. It projects to lots of other brain areas from where it's found at the third ventricle. Okay, and our next CVO is the median eminence of the hypothalamus. So just like the name implies, this is a region of the hypothalamus, specifically the median eminence, and it lacks a blood-brain barrier. If you look at this drawing on the slide here, we've already talked about the OVLT found right here and the SFO found up here. And we've also talked about the area prostrema, which is found down here near the midbrain and the cerebellum. Well, right here is the pituitary gland and the pituitary stalk, and so the hypothalamus is right up here. And its median eminence or medium side has no blood-brain barrier. And that's important because this is how the hypothalamus releases hormones into the vascular system so they can travel to the pituitary gland. In the endocrinology videos, I talk in detail about how the hypothalamus controls the pituitary gland by releasing its own hormones. Well, those hormones have to get beyond the blood-brain barrier, and they do that by being released from the median eminence of the hypothalamus. This allows the hypothalamus to regulate the pituitary gland. And then finally, a couple of other areas that don't have a blood-brain barrier that you should be familiar with are shown on this slide. So first of all, the posterior pituitary gland lacks a blood-brain barrier, and this allows it to release oxytocin, an antidiuretic hormone. Recall that the posterior pituitary gland is kind of like a big neuron. It's different from the anterior pituitary gland, which is more like an endocrine organ. So the posterior pituitary gland has to have ability to release its hormones directly into the bloodstream, and therefore it lacks a blood-brain barrier. And then the pineal gland, which I talk about in other videos, releases melatonin. It also lacks a blood-brain barrier so that it can secrete melatonin into the bloodstream. Okay, I'll finish this video by talking about cerebral edema, which refers to fluid accumulating in brain tissue. This can be life-threatening because it can increase intracranial pressure. So this can lead to symptoms like headaches, papilledema, which is something you can see in the optic nerve on fundoscopy exam, potentially herniation of the brain, which I talk about in other videos. And we broadly divide cerebral edema into two subtypes based upon the mechanism by which the edema occurs. Those two subtypes are vasogenic and cytotoxic edema. So let's talk about those now. So cytotoxic edema is the form of cerebral edema that occurs when the blood-brain barrier is intact, but sodium pumps have failed. So sodium pumps require energy to work, and so if there's some problem with metabolism in cells in the brain, then sodium pumps can fail, and this can lead to the accumulation of sodium inside of cells in brain tissue. Water then follows sodium into cells, and you get cellular swelling. So what are some clinical situations where you can see cytotoxic edema? Well, first of all, this is the classic form of cerebral edema to see early in patients who have stroke or ischemic brain damage. Usually within the first 24 hours, if a stroke patient has edema, it's going to be cytotoxic edema. This is also the type of edema you can get in patients who have severe metabolic derangement. So for example, patients who have severe hypoglycemia and develop cerebral edema, that can be cytotoxic edema. You can also see this when patients have severe sodium disorders or hyperammonemia from liver failure. All of these things basically can disrupt cellular metabolism in cells in the brain, and that can cause the sodium pumps to fail, and that leads to cytotoxic edema. This is often diffuse edema, although it can be localized, but obviously if you're going to get edema from a glucose disorder, it's probably going to be diffuse. And there are findings on MRI that can help you distinguish cytotoxic edema from other forms of edema. But they're not going to ask you this on step one. It's far too advanced. But these are some pictures on the slide here, just so you know what it looks like, cytotoxic edema. On certain imaging sequences, it will light up like what's shown in these images. The other type of cerebral edema is vasogenic edema. This occurs when there's a breakdown of the blood-brain barrier. This allows fluid and proteins to go into the intravascular space, and you get swelling of brain tissue generally. So this isn't just cellular swelling like you see in cytotoxic edema. This is generalized brain tissue swelling from leakage of fluids and protein. And this is the type of cerebral edema you can see in patients who have trauma. Obviously, trauma can disrupt the blood-brain barrier. Tumors growing in the brain can also disrupt the blood-brain barrier. If there's inflammation in the brain or spinal cord, like in meningitis or encephalitis, this is the kind of edema you can get. And then you can also see this type of edema in stroke patients, but it's a late finding, usually after 24 hours. So it takes some time for the ischemia to cause breakdown of the blood-brain barrier. If there's early swelling and stroke, it's going to be cytotoxic. If there's later swelling, it's going to be vasogenic. And this, in contrast to cytotoxic, can sometimes be localized depending upon the cause of the vasogenic edema. And this is another example of vasogenic edema that's localized to the posterior portion of the brain. Once again, they're not going to expect you to diagnose cerebral edema by imaging. I just want to show you this so you have some idea of what it looks like. I'll finish this video by talking about hyperosmolar agents, which is the type of drug that mannitol is. Mannitol is the classic hyperosmolar agent. And what mannitol does is it increases plasma osmolarity. And when you do this, you're going to draw water out of cells and tissues. And so for this reason, sometimes mannitol is infused in patients who have either cerebral edema or glaucoma. The reason for this is it's going to draw fluid out of brain cells and tissue beds. The fluid's going to go into vessels and then go out in the urine. And this is going to lower the intracranial pressure. It can also lower the intraocular pressure in patients who have glaucoma. And sometimes mannitol is called an osmotic diuretic because it increases urine output. Although really, we never use this drug just to increase urine output. The main times we use it are what are shown in this slide to treat cerebral edema and glaucoma. Now, when you infuse mannitol, not only do you draw fluid out of cells and tissue beds into the vascular space, but mannitol also briefly disrupts the blood-brain barrier. What it does is it draws fluid out of endothelial cells so they shrink, and this disrupts those tight junctions that are part of the blood-brain barrier. This is a temporary effect and it resolves after about 30 minutes. But this is important sometimes in treating patients with chemotherapy who have brain tumors. If you give mannitol just before you administer the chemotherapy, you briefly disrupt the blood-brain barrier, and this allows the chemo agents to reach brain tumors. And so this can come up in your exams, the issue of giving mannitol prior to chemotherapy. The reason this is done is to temporarily disrupt the blood-brain barrier. And that concludes our module on the blood-brain barrier.